Huntington's disease is a neurodegenerative genetic disorder which was first described by George Huntington in 1872. It affects muscle coordination and leads to cognitive decline and the early death of those who have the disease. Complications such as pneumonia, heart disease, and physical injury from falls reduce life expectancy to around 20 years after symptoms begin. There is no cure for Huntington's disease, and full-time care is required in the later stages of disease. It affects both men and women equally and typically reveals itself around the middle age of its patients. It affects around 30,000 people each year, with at least 150,000 others having a 50% risk of developing it. It is caused by an autosomal dominant mutation in either of an individual's two copies of the gene Huntington. The gene interacts with proteins which are involved in transcription, cell signaling, and intercellular transporting. It also acts as an anti-apoptotic agent, preventing programmed cell death, and controls the production of brain-derived neurotrophic factor, a protein which protects neurons and regulates their creation during neurogenesis. It is thought that the disease is not caused by inadequate production of HTT, but by gain of toxic function of the mutant protein. All humans have two copies of this gene, which contain a trinucleotide CAG repeat that has a 9 to 36 threshold for normal individuals. When this threshold is crossed by exceeding the 36 repeats, a mutant form of this protein is produced, which causes a decay of neurons, which are the cause of pathological changes, and which in turn cause the disease symptoms. If the amount of repeats exceeds 40, then the disease will be fully penetrant. It is an autosomal dominant disease, meaning that the disease will almost always show up in individuals who have a copy of the disease, meaning that it is fully penetrant. Offspring of those with the disease have a 50% chance of obtaining the disease themselves. In the rare situations where both parents have an expanded HD gene, the risk increases to 75%, and when either parent has two expanded copies, the risk is 100%, meaning all children will be affected. Individuals with both genes affected are rare. In order to test for Huntington's disease, I would use transgenic mice as a model organism. These transgenic mice are produced by implanting transformed embryonic stem cells in a surrogate mother who in turn produces chimeric offspring. These offspring will then produce the transgenic mice of interest. They would have the 5' end of the HD gene with the appropriate promoter, exon sequence, and an excess of 130 repeats. If the transgene is expressed at both the mRNA and protein levels, then the mice would exhibit the symptoms of the disease, including involuntary movements, tremor, seizures, and non-movement disorder components.